Dr. Olawoye is a microbiologist and bioinformatician, took me a while to get that one, with particular interest in understanding bacterial pathogens, their evolution, as well as antimicrobial resistant patterns. To do this, he uses computational tools to analyze genomic and epidemiological data to provide insights to public health agencies. An interesting fact about Idowu is that he is ambidextrous, which is a trait that he shares with only 1% of the population. So without further ado, Dr. Olawoye.
Just a few years ago, I completed my PhD studying the bacteria that causes tuberculosis, which in fact is a slow-growing bacteria. It's hard to grow in the lab and experiment on because it's highly infectious and it can be drug resistant. But now it's time to move on. And when microbiologists like myself switch to studying new bacteria, they would either study a new bacteria in a different bacteria that belong to the same family, or switch to something completely different, maybe something easy where they can grow in the lab and it's fast growing and they can plan experiments from day to day, which would lead to scientific discoveries and obviously scientific publications. But I've done the complete opposite of this. I've decided to study something that is difficult to grow in the lab, is equally transmissible, and it can be drug resistant. Allow me to introduce to you MGen, which is also known as Mycoplasma genitalium. This is a sexually transmitted infection that can infect both male and female, and it disproportionately affects the marginalized groups. And this bacteria, if left untreated, can actually lead to things like infertility or even cervical cancer. And you might be wondering, why have I decided to take this route? Like, why did I decide to study something that's hard to grow? And that's because I'm trying to catch it before you or someone else catches it. And because it's an important bacteria, we need to be able to test it in the lab. And it's quite daunting. It's a difficult, thing to do. One of the challenges is because it's extremely slow growing, which means most bacteria usually take a day to grow in the lab. A slow grower like Mycobacterium tuberculosis will take about 6 to 8 weeks. But for these, will probably take about 2 or 3 months if you're lucky. Also, it is a fastidious bacteria, which means it's very picky. Imagine you have a toddler who's picky with food. That can be quite frustrating. And it's the same with this bacteria. You need to replicate or mimic the human host conditions for it to grow, which can be quite challenging. And also, it's resistant to most antibiotics that are used in treating this disease, and that's because it lacks a cell wall that most bacteria have. Because it doesn't have this, these drugs will not target what's not present. And like I said, it's a very important bacteria. It's a very important disease. It's not quite known. It's understudied, and that's because it's really hard to study. It's hard to grow. But with the current trends, there are more infections spreading which are difficult to treat. And one of the detection methods is done using PCR machines, which is an equipment like this. It's a bent-up equipment like this, and it usually will require additional equipment like a computer or gel imaging device, and for additional analysis. And it uses variable temperatures to amplify the DNA of the bacteria to know if it's present in your clinical sample. And it can usually take about 2 to 3 hours to get your data, which can be quite long if you're not really skilled to do this kind of analysis. And it's pretty expensive also, which means labs in low-income countries or under-resourced area would have challenges in procuring this. So what my research is doing is to take a new technology called LAMP assay, which doesn't require the large equipment and all the secondary devices for analysis. What you just need is a heating block like this. So you can literally procure these on eBay. And what it does is it allows for colorimetric detection, which means you can visualize the result with your eyes without needing computers or anything. And it uses a constant temperature to amplify the DNA of your bacteria. And your results can be ready under an hour. And it's cheap and easy to use, which means we can deploy this in under-resourced areas. And this is what your result will usually look like when you have a PCR analysis. You could have graphs like that might be quite difficult to interpret, or a gel image of that nature. But with what we've been able to do in the lab, this is the results we achieved. You can see there's a color change from red to yellow. Yellow is where we have a positive sample to MGen, and the red one is a negative control, which means you can literally see the change if the sample, if it's positive or not. And the overarching goal of my research is to improve STI testing. And because we know time is of the essence, we need to be able to test drug resistance rapidly, which will then prevent the progression of this disease to its chronic stage and improve health equity to everyone. And I would like to appreciate our team and our founders, and thank you for listening.